All right, welcome to our first annual Fatty Acid Symposium. My name is Tavis Piatoli. I am the healthcare account manager and oversee our sales department at OmegaQuant. So I will be your host. We'll cover 20 years of the omega-3 index and what lies ahead. That'll be presented by Dr. Bill Harris, and we'll get to that shortly. As we're getting started too, I want to thank our co-partners. We do not have sponsors for this event. Also, if you want to let us know where you're from, put it in the chat. It's always good to see all of our attendees and where they're located from all around the world. So let me share my screen. I want to thank our co-partners. We do not accept sponsors, so this is a completely non-commercial event. There are no sponsors for this event, just co-partners. These are individuals we work closely with, and also they share a lot of our resources to their followers as well. Carlson Labs, Vivo Mega, Nutraceutical World, Nordic Naturals, also OmegaQuant, Yoli, and GoEd Omega-3. So we want to make sure we thank them and also for their support in helping share these resources. So Dr. Harris, he's been the leading researcher in omega-3 fatty acid field for over 40 years with more than 360 scientific papers on fatty acids and health, the vast majority on omega-3. He has been on the faculty of 3 medical schools, the universities of Kansas, Missouri, at Kansas City, and South Dakota, and has also received 8 NIH grants to study omega-3s. He's the co-author on 3 American Heart Association papers and statements on fatty acids and heart health, as the co-inventor of the omega-3 index and other omega-3 blood tests, and founder of OmegaQuant Analytics. Dr. Harris has been ranked among the top 2% of the scientists worldwide based on his impact on the research. So, Doc, thank you so much for being here today, and I'm going to turn it over to you.
Great, Tavis. Thank you very much, and welcome everybody to what hopefully will be a fun series of 10 very good lectures this week. I'm excited to be able to watch them and do a couple of them myself. We thought we would start off with some history, and so I'm going to talk about how the omega-3 index came to be and what's happened to it over the last 20 years since it's been actually in use. And so let me see if this will go forward. I can advance this maybe. There we go. So I thought I'd start with really the birth of the omega-3 story itself. And this is a paper we published in honor of Dr. Bang and Dr. Dyerberg, who are really the 2 discoverers of the omega-3 story back in Greenland back in 1971. And their first expeditions over there revealed that the Eskimos in the particular area they were working seemed to be sort of immune to acute myocardial infarctions. They discovered after almost over a 7 or 8 year period that there were omega-3s that were in their blood and in their food that was responsible for the benefits they saw. Since that time, the amount of research that's been done on omega-3 has been kind of pooled together here. This is a list of really the top molecules, drugs, nutrients, whatever, in medical history that have been studied. And you'll notice here penicillin here is not a lot of research recently on penicillin, but originally there's a ton of work on it. But then vitamin D, this is the number of papers published on these different topics. And you get down here to folic acid and omega-3, and they kind of are tied for 5th place in the total amount of research. And we've known about all these other drugs and nutrients for a long, long time. We've really only known about omega-3 since the early 70s. So we haven't had nearly as long to generate research, but a lot of work has been done on omega-3, certainly more than on Valium or even statins. There's been more research on omega-3. So it's a major focus of research around the world. Okay, well, I promised we'll talk about 20 years of the omega-3 index. It's actually, as of today, 21 years, okay, but we'll call it 20 years because we published our first paper in a journal called Preventive Medicine in 2004. The title of the paper was the Omega-3 Index, a new risk factor for death from coronary heart disease. And we put a question mark behind it just to be kind of cautious, because it was a proposal that this would be a new marker. I was first author, Clemens von Schacke, my colleague over here on the right, a cardiologist from Munich, Germany. We kind of came up with this idea together, published this paper together, did research together, and certainly want to acknowledge his long-term contribution to not only the creation of the omega-3 index, but his own research in the omega-3 story. He's doing a great job. So obviously we need to talk about what is the omega-3 index. It's a measure of the amount of EPA and DHA in red blood cell membranes. It's expressed as a percent of the total fatty acids. So in this cartoon or cutaway of a red blood cell, which is, of course, like every other cell, it's a lipid bilayer, outer layer, inner layer. And in this particular cartoon, we have 64 fatty acids. That's the little dangly things, or dangly up, dangly down things, or fatty acids. Each ball represents a phospholipid with 2 fatty acids. And this cartoon, we have 4 omega-3 fatty acids out of 64 total fatty acids. So it's pretty simple math. 4 omega-3s out of 64 total is 6.25%. So that would be the omega-3 index of the person from whom this red blood cell was taken. And that's a pretty simple way. It's very much like in a sense, like a hemoglobin A1C metric. It's expressed as a percent. It's measured in a red blood cell. It's a reflection of long-term omega-3 status, like A1C is a reflection of long-term glucose status. They're a very similar concept. How do we measure the omega-3 index? We do it with a method called gas chromatography, a very classic analytical methodology in chemistry, analytical chemistry. Without going through all the steps, I'll just show you what's called a chromatogram, a picture that we get out of our instruments when we analyze a red blood cell fatty acid composition. Each one of these peaks in this tracing is a different fatty acid, and they're labeled here. This first one down here is myristic acid, and this is palmitic acid. And then we have 18O, which is stearic acid, oleic acid, linoleic acid, the omega-6, which we'll be talking about actually in the next program or next talk on this Monday of the symposium. Arachidonic acid is down here. Actually, I should say the bottom axis is time. So it takes time for these samples to be analyzed. Right here under the big EPA is a little bump called C20 with 5 omega-3. So that is EPA. This is not blood taken from somebody that's taking much fish oil, I'll tell you that. It's pretty little bump. And then over here is DHA, a much bigger bump. And what we do is we measure the area under the curve of each one of these fatty acids. We add it up, and then we decide what percent of the total area is made up by EPA and DHA. And that's the omega-3 index. So it seems simple, but there's a lot of work that gets in actually producing a picture like this. So what actually was the reason we came up with the omega-3 index? Well, there were 2 studies that together inspired both Dr. Bonshaki and myself. When we heard these studies presented, the light bulb went on, and that was about 2003. There had been a study first done by David Siskovic and his colleagues in Seattle, Washington, published in 1995. This was published in JAMA, and this was looking at the relationship between omega-3 levels. It was actually red blood cell EPA, DHA. They didn't call it the omega-3 index, but that's the metric that they used in this study. And it was a cross-sectional study, meaning this study was drawing blood from individuals who were suffering primary cardiac arrest. When the ambulance showed up and people were down on the sidewalk having had a heart attack, the paramedics would collect a small blood sample and they saved it. And they then compared the omega-3 levels in the blood of people who were having heart attacks at the moment with community-based controls, people who were not having a heart attack at the moment, otherwise age and sex, race matched. And what they found was that if you look at the quartiles of the omega-3 index and looked at the risk for being actually the odds of being a case in this situation was somebody having a heart attack, the individuals who had the highest omega-3 levels were basically 90% less likely to be a case compared to people who had low omega-3 levels. So this was cross-sectional again, and it was statistically significant. It pointed to a potential relationship between omega-3 levels and risk for sudden cardiac death. But at a cross-sectional study, you really can't come up with risk. All you can do is look at odds for being a case, which led to this study. This is a prospective study done by Christine Albert, now in Southern California at Cedars-Sinai, a cardiologist working, doing research there. She did this work when she was at Harvard, and she looked at the blood omega-3 levels, essentially the omega-3 index in people, and then in the physician's health study. And they had saved blood samples at enrollment to the study. When the people enrolled in this study, they were healthy. They didn't have any known cardiovascular disease. And then the samples were frozen away. And then after some 17 years, roughly 100 or 100 and some people had died of sudden cardiac arrest. And so the researchers pulled out the blood samples from baseline from these people and compared it to blood samples from others who were matched controls who had not had a heart attack and died. And they found exactly the same thing Dr. Siskovic and his colleagues found, that the people who had the highest omega-3 levels out here on the far right, the highest quartile, 25% in the highest group, had the lowest risk for, in a prospective way, developing cardiovascular, fatal cardiovascular disease. So this was good confirmation that the omega-3 index actually predicted important outcomes. So that's what prompted Dr. Von Schacke and I to say the omega-3 index, the omega-3 levels really are clinically meaningful and should be available to physicians to take care of their patients because it's a strong risk factor and it's a very modifiable risk factor. We knew that as well, obviously. And so when we proposed the omega-3 index, we had to come up with target values. If it's going to be a risk factor, you've got to say, well, what's a good level? What's a bad level? To do that, we used the data that was available at that time, which is not very much actually. But we estimated what the omega-3 index was in a variety of studies that had been published, doing our best to figure out what the levels had been, even if they didn't measure them. They gave us some idea. And we then basically just took the average of all of these values, and it came out to be 8.1%. So we said 8.1% seems to be a good target based on the protection from cardiovascular disease. That's turned out it's been a great target for just about everything we look at. And on the left-hand side, what's a bad place to be? Under 4% looks like to be the place where you don't want to be. And so we have the scale basically saying 4% and below is we'll just call it undesirable, high risk level. Then there's an intermediate level between 4 and 8. And then there's the desirable 8 to 12% is the place we think everybody ought to be if possible. This is a fairly typical level for older Japanese, not the younger Japanese who are adopting, unfortunately, Western dietary habits. But a lot of research in older Japanese showed this is their level. Typical levels for the U.S. and Europe are somewhere down here in the intermediate zone, 5.5. People we see who have levels under 4%, we've seen it in vegans, and we've seen this level in a Palestinian population we researched, and also in U.S. military personnel deployed in Iraq, sadly, have levels under 4%. So that's the extreme in that end. Recently, Jean-Philippe Schuhart and our group worked together to come up with a world map of the omega-3 index. And we did our very best to find in the literature data published since 2000 on blood levels of omega-3 fatty acids on a countrywide basis. And we tried to put this map together to color it. The different colors are here. Red is it's under 4%. And there certainly are some countries where it appears that there is especially high need for omega-3. I would point out that all the gray are places where we don't have good data on omega-3 status. So there's a lot of work yet to be done. The green, of course, is the highest omega-3 levels, and as you would expect, Nordic countries, Greenland, Alaska, Japan, Korea are places where seafood intake is very high, and their omega-3 levels reflect it. In the United States, North America, pretty much in that 4 to 6% range. Central Europe, also in that level. France and Spain are a little bit higher. Anyway, you can see, get an idea here that the omega-3 index is generally below optimal for the vast number of people in the world. Now, of course, I said the omega-3 index is measured in red blood cells, and I want to talk a little bit about why that is and what evidence we have that that's the right way to do it. So why do we measure the omega-3 in red cells instead of plasma? There are some laboratories that measure plasma omega-3. I'll show you here some of the evidence for why we think the red cell is a better marker. Here's a study we did back in 2010, and we looked at the within-person variability, like you draw blood samples, multiple blood samples over time, does the level of the marker stay the same? And it should, if everything about that person's dietary habits is remaining the same, then the blood level ought to stay the same. So we had 20 healthy volunteers. We tested them after an overnight fast weekly for 6 weeks. And then we measured the plasma EPA DHA concentration, and we measured the red blood cell. So this is the omega-3 index. And as you can just see, each one of these is an individual. The amount of week-to-week noise is much higher in plasma than it is in red blood cells. This is why it's kind of like a hemoglobin A1C for omega-3s, because it's a much more stable marker. The total what we call a coefficient of variation for the red blood cell metric was about 4%, I mean within person, compared to the plasma level, which was roughly 16%. So that's this and this. So actually the omega-3 level in red cells is much, actually in large populations, you could probably use plasma or red cells, but dealing with any one person as a patient, it's best, we think, to use red blood cell because that's going to give you the most accurate look at their status. We've also looked at the effects of fasting on the omega-3 level in the blood. And again, another study with 20 healthy volunteers, we tested them before and after 4 hours after they were given what we'll call a hearty breakfast, a good McDonald's breakfast. And what we found was that the omega-3 level in plasma significantly decreased in the people in the fed state after they've had the meal compared to the fasting state, but there was no significant change in the red blood cell level. So, why did the omega-3 level go down? It's not because they weren't eating fish so much. It's because when you eat a meal with fat in it, and the fat is then in your blood, the fat, non-omega-3 fats, the saturated fats, monounsaturated fats, other omega-6s, their levels go up in the blood. And because of that, the omega-3 levels go down as a percent of total. So that's what we see here, that you don't have to fast when you're having your red blood cell omega-3 levels tested. They're not affected by fasting. Another important, I think, reason to use red cells is because plasma levels get perturbed by an acute dose of EPA and DHA, but red cell levels do not. So if you have a big fish meal the day before your blood's drawn, or if you take a big omega-3 supplement, here we took, again, 20 volunteers, gave them 4 grams of the omega-3 pharmaceutical product, Lovesa, Omicor in some parts of the world. We gave it with a meal, and we then tested their blood omega-3 levels for the next 24 hours. And if you look at the change in plasma EPA, and this is as a percent of total fatty acid, it goes up after you take the pills, as it should. And if you look at plasma concentrations of EPA and DHA, and in micrograms per mil as opposed to a percent. That definitely goes up. But if you look at the red blood cell levels of EPA and DHA, they're completely immune, fundamentally, to any acute dose of omega-3. So again, it's a better marker of overall uh, status of omega-3, regardless of whether you had another meal, regardless of whether you've had an acute load of omega-3, which could trick it. The omega-3 index is it's not easy to measure, and there are certain analytical things one has to do it right. We early on in the development of the omega-3 index, we had a comparison to multiple other laboratories that were offering some kind of a red blood cell omega-3 test. So we took blood samples from, actually, this is probably from me, and we sent them to 4 other labs besides OmegaQuant and had them run the omega-3 test. And what we found, so this was a 10% or roughly 10.5% omega-3 index for me. And these other laboratories, which we won't need to name them, but one of them gave a 14%, one of them gave a just about a 4%, one gave a 6, and another one gave about a 9. So the amount of, you can't necessarily trust the omega-3 index that's given from another lab. So the important thing is, if you're going to test your omega-3s, always use the same lab because you don't want to go lab to lab because you can't necessarily compare them. We think we do the best job on it, but that's just our opinion, of course. Let's go on and look at the question, does the omega-3 index reflect tissue levels of omega-3? And I think that's shown nicely in the study by Jim Metcalf in Australia. This was now almost nearly 20 years ago. They did a very nice study looking at giving fish oil to people who were awaiting heart surgery, basically. And it took quite a while for some people to actually get into the surgeon. So there was quite a span of time. So they took blood and they took heart tissue, what's called the left atrial appendage. It's a little piece of the heart that can be sampled and removed without any damage. So they collected it on 28 patients. All these patients took 6 grams of omega-3 per day between 10 and 70 days before they had their open heart surgery. And so that spread out the time very nicely. Then they looked at the rate of incorporation of EPA and DHA into the red blood cell and into the heart. And in this graph over here, it shows you at the bottom, the duration of fish oil supplementation, the number of days that it was taken. And here's EPA and DHA as a percent of total fatty acid. So this, for the red cell, that would be the omega-3 index. And so what they showed here is the solid line is the heart, the rate at which the heart is accumulating omega-3s over that period of time. And the dashed line, erythrocytes, red blood cells, this is the rate that the red blood cells were accumulating at. So fundamentally, they're accumulated at the same rate. And what happens in the red cell is what's happening in the heart. And there's other evidence that we've got that this is true, but this one slide makes the point that for at least major organs, I would say outside of the brain does not, it's a level, omega-3 levels are not nearly as malleable as other tissues. But there are a lot of other tissues that are important too. So another point I want to make here is supplementation, response to supplementation cannot be predicted with accuracy. And I say that, I'm sorry that's the case, but that is the case. We can't look at say, if you take a certain amount of omega-3, your omega-3 index is definitely going to change by this much. And the reason we say that is from the study conducted at Penn State by Dr. Mike Flock and his colleagues. This was a study where they took about 25 people in each group. So about 125 people were in this study, all healthy volunteers. They were given either a placebo or 300 milligrams of EPA DHA a day, 600, 900, or 1,800 milligrams of EPA and DHA a day, and they did it for 5 months. So this is plenty of time for the red blood cells to all incorporate omega-3. And as you can see, on average, there was a dose response curve. The more omega-3 in each one of these dots is an individual person's response. But on average, people went up as they should. The omega-3 level kept going. Omega-3 index went up as the dose went up. But the important thing here, I think, is to look at the spread of the change. What we're looking at is the change in omega-3. Um, and it's not just the actual level. So the actual level started out about 4.3%. So the people in the placebo group didn't change. The people in 300 group, it went up in fairly tight response. But if you look here at the 1800 milligram a day, and we know these people took their supplements because we did pill counts afterwards. Some of the people changed by almost 8%. So they went from, say, 4% to 8%, for 12%. So it's a delta of 8%. Others only changed by maybe 1%. Remarkable variability. And the whole point of this, I think, is that you've got to measure it if you're going to know it. You cannot estimate your own omega-3 index just from knowing how much omega-3 you're taking. Everybody is different. There's a lot of different factors that play into what causes the omega-3 to go up and down. I guess I need to pick up my speed here. We have been interested in how much omega-3 you need to change your omega-3 index. When we did a study, Dr. Jackson, Christina Jackson, was the first author on this paper, and we looked at we surveyed about 3,500 people who had been who had turned in red blood cell, or rather dried blood spots, to OmegaQuant for analysis. And we asked them how often they ate fish meals per week. And each one of these pairs of bars is a higher amount, from no fish to 3 times a week. And then we also asked them if they took a supplement or not. No supplement is light blue, and dark blue is if they, yes, took a supplement, fish oil supplement. We don't know how much, or which kind of supplement. We just yes or no. And then we look at the omega-3 index of these groups, and you can see that if you just look at the light blue bars, people who don't take omega-3 supplements, as their fish intake goes up, their omega-3 index goes up, and that makes perfect sense. And if you're taking an omega-3 supplement and eating fish, then your omega-3 index goes up in the same stair-step fashion, which documents really the reliability of the omega-3 index metric. What levels were achieved? Well, these are error bars. This is the standard deviation. So there is a lot of variability, even in people who are not taking supplements. Their average omega-3 index was 7.1%. So you can certainly get an 8% omega-3 index just eating fish. It's just easier to do it if you are taking a supplement. So these people are the ones, the folks that at 3 times a week eating fish and taking a supplement had on average an omega-3 index of 8%. I have pointing out this particular box here. This is eating fish twice a week and not taking a supplement. That is fundamentally the standard American Heart Association recommendation for fish and omega-3 intake. This will not get you an omega-3 index of 8%. So that's not adequate to do it. The only group, as I said, that got an average omega-3 index of 8% was those taking a supplement of some kind and eating fish 3 times a week. So there's a lot of variability in what it takes, but it's good to know this can be done with diet alone. It doesn't have to be done with fish oil supplements, but it's easier to do with fish oil supplements, I think. So, interesting. What affects the omega-3 index response to a supplement besides just the dose of omega? Obviously, the dose of omega-3 makes a difference. If they eat fish regularly, if you have a higher baseline omega-3 level, because you eat a lot of fish, then taking a supplement will typically not give you as much of a rise because you're already near the top of the curve. So that can affect your response. If you're not eating much fish at all and you have a really low omega-3 index, the same dose of omega-3 will raise your levels higher. What form is EPA and DHA in? If the supplement is an ethyl ester form, they're typically less well absorbed than the triglyceride forms, particularly if the ethyl ester form is not taken with food. If you take ethyl esters with food, that certainly helps with their absorption, but they're still not as well absorbed as triglycerides. taking them with food, as I just sort of alluded to, even the triglyceride form is better absorbed if it's taken with food, more acutely absorbed. So that it's always important to take supplements with food, ideally with food with some fat in it, not just carbohydrate, so you can get all the lipid digesting enzymes secreted with the meal. And fitness, this is sort of an interesting one. We've seen that intense exercise appears to decrease the omega-3 index. Particularly high-end athletes are seeing fairly low omega-3 index levels, even if given omega-3 standard supplements. So there's a lot of research that needs to be done on how to get really fit athletes higher levels of omega-3. So that's a kind of a background on the omega-3 index and what affects it. Now I'm going to kind of shift over to some epidemiology and look at the data we have where the omega-3 index has been shown to be a predictor of a variety of important outcomes. Mortality, that's death from any cause. Cardiovascular disease, particularly, which is where it started. Interesting research on dementia and Alzheimer's disease, COVID-19 studies we've done, and a new paper just last week where we saw a relationship with sepsis. So let's go through some of this. First, I wanted to point out that this is a study that we did within what's called the Women's Health Initiative, where we measured the omega-3 levels in this was done in Framingham, not WHI. In Framingham, offspring cohort, 2,500 people free of cardiovascular disease at baseline, average age 66. We measured their omega-3 index at the time of the beginning of the study. And then we followed them for about 7 years. And we looked at who died of what during that 7-year period. And we ranked, this is based on the omega-3 index. We ranked people from the lowest omega-3 index to the highest omega-3 index. And you see that the higher the omega-3 index goes, the lower the risk of dying. So the omega-3 index was a predictor of risk for death. I should say that the median omega-3 index in this top quintile was around 7.7, so pretty close to 8%, as we originally thought. What we thought was interesting was to ask the same question of blood cholesterol levels, known to be a risk factor for heart disease, and we thought for death. But we turned out when we take people and rank them not by their omega-3 level, but by their cholesterol level, the people with the lowest cholesterol serving as the reference group, and then as cholesterol levels go up over 218 in this group, you can see that there is as cholesterol levels kind of go up, the risk for dying seems to go down. It's not statistically significant here. And this trend is obviously not significant because people at the highest end of cholesterol and the lowest end had the same fundamental risk for death over the same period of time. So the point is not that cholesterol is useless. That's debatable. We think people should still know their cholesterol level, but if you're interested in your cholesterol, you should be more interested in your omega-3 index because it's a better predictor of this major health outcome, death, than cholesterol is. In a similar vein, we did another study within the Framingham cohort asking different questions. And this time we were looking at the relationship between the predictive ability of the omega-3 index predicting risk for death over time, and we compared it with whether people are smoking or not. So smoking is a well-known risk factor for a variety of health outcomes. So we asked the question, we looked at groups of people who had a high omega-3 index, meaning the highest quintile, and they were not smoking, high omega-3 who were smoking, and then people in the lowest omega-3 group not smoking, and the lowest omega-3 group that was smoking. And this is the theoretical survival curve based on our data observed. This is observed actually, but using a statistical model. People who are the worst off, who most likely to die over this roughly 10-year period, 57% of the people were, rather, I guess you'd say 43% of the people died who were in this group, which is low omega-3 and smoking. So that makes some sense. Roughly 50-50 chance of living 10 years if you're from age 65, if you're a smoker and have a low omega-3. The best situation was here, the people who were non-smoking and had the highest omega-3s. Only 13% of them died. over that period of time. And the two mixed groups had virtually the same predicted death rate, around 25% death rate. If you were a smoker and didn't have high omega-3, or excuse me, if you were a smoker and you had a high omega-3, or for a non-smoker and a low omega-3, basically they were about in this category. So the point is really not that you can take omega-3s and keep smoking. That's certainly not the message. The message is omega-3 levels are an important risk predictor. If you think smoking is an important risk factor, you should think a low omega-3 is an important risk factor as well. So these are two studies that we did in the Framingham cohort. We have since branched out, and this is a study that I was able to lead with a large consortium of other cohorts like Framingham. We actually had 17 prospective studies in this research project done with a group called the FORCE. It's a FORCE coalition. FORCE stands for Fatty Acids and Outcomes Research Consortium. So this is a group of many different researchers who have cohorts like Framingham. A cohort is a group of people that you measure their status at the beginning of the study, and then you follow them over time from all around the world. And we were looking here at the relationship between blood omega-3 levels and risk for total and cause-specific mortality. So I'll explain that in a second. In this study, we had 42,000 individuals involved from 17 different studies in 10 different countries. Very broad look, really worldwide look at the omega-3 relationship with risk for death. Overall, they were around 64 years old, about half were women. The median follow-up time, meaning the time between blood sampling and when the end of follow-up, which is roughly 16 years, median follow-up time was 16 years. And so we're sort of looking at death that occurred between roughly age 64 on average, and with 16 years of follow-up, years 80. So in that window of time, who died and what they die of. We had almost 16,000 deaths out of these 42,000 people, which is a large, huge sample size. A third of them, or 30%, were cardiovascular, 30% were cancer, and about 40% were other causes. And so what did we find? Yeah, what we were able to do is we would take all the people that we had, all the blood measures, and rank them from the lowest omega-3 level, the lowest 20%, and this is the highest 20%. So this is called a quintile analysis. And so we're looking here at the hazard ratio, which is the risk of dying. And this is all-cause mortality, so death from anything. And you can see a very nice dose-response relationship. The higher the omega-3 level, the lower the risk for death, down to about a 20% lower risk in the highest omega-3 group compared to the lowest omega-3s. So that's very impressive. We also, as I said, looked at cardiovascular disease, cancer death, and death from other causes, and the same pattern is seen with all three. So it's not just that the omega-3s are protective in cardiovascular disease. They're seen to have a protection against cancer. They seem to have a protection against really death from all causes, because your body is just healthier all over, everywhere. And so this is compelling evidence. We estimated what the omega-3 index was, because not all these studies measured the omega-3 index. Some of them measured plasma, and we had to convert that. But the estimated omega-3 index in the lowest quintile was about 3.6%, which fits our under 4%, and the highest in quintile 5, and these people are not taking omega-3 supplements, okay? This is dietary, almost completely. 7.8% was the mean value in this quintile. So again, more evidence that the 8% target is a very reasonable target for overall health. So that's the mortality side. Let's just look at specifically some cardiovascular outcomes. This was one of the first studies we did after we came up with the omega-3 index, actually published in 2008. We looked at 768 cases of acute coronary syndrome, people admitted to the hospital with an acute coronary syndrome, fundamentally a heart attack or unstable angina. and we drew blood when they were admitted, and then we found control people who were also coming to the hospital, not with a heart attack, but just to come to have their blood drawn before they go to a doctor visit. So those were our cases and controls, and we separated people into omega-3 index under 4% and over 8%. And we found that people who had a omega-3 index over 8% were about 70% less likely to be an acute coronary syndrome patient. So that's the way you do these cross-sectional studies. We adjusted for tremendous number of other potential variables that might explain this difference, and none of them did. So it's the omega-3 level that was high that was protective. We also did a meta-analysis of 10 studies. This was long before we did the 17 study project, 27,000 subjects. And what we found here, if you compared to an omega-3 index of 4% or below, those who had an omega-3 index of 8% or above, 35% reduction in risk for death. And so it fits there with again, what we proposed is a good target. This is an interesting study. I think this is the background for it, but we're still in the cardiovascular world. So this is a study it's a randomized trial. This is not observational. This is a randomized trial that was done by Ray Kwong and his colleagues at Harvard. They gave 4 grams a day of Loveza for 6 months to people who had a heart attack. And then they looked at using cardiac MRI, looking how the heart muscle healed over the 6-month period with and without omega-3. And their primary outcome is what they call the left ventricular end systolic volume index, which is a mouthful, but it's this thing here. And they showed that the people who had the biggest change in the omega-3 index, so this is the highest, this group here, the people that changed the omega-3 index the most, had the greatest reduction in this metric, which is good. Healthier down is a better outcome. Their heart was healthier and was healing better. And so we published this study back in 2016. The omega-3s were helpful, and it looked like there was a relationship between the final omega-3 index and the health outcome. Again, the higher group had 11% omega-3 index. Now, these people were supplemented, so that's why the levels are so much higher than we saw in the observational studies. Last year, Dr. Kwong and his colleagues went back and looked at what happened to the patients from this study, because they were only treated for 6 months. So here's the 6-month treatment period after they'd had their heart attack, and then the study was stopped, and there was no more treatment. And so they went back and looked at the evidence or the data, what happened to them, and they found that based on what they call intention to treat, meaning how they were assigned to a different group, green and blue, placebo, and omega-3, the rate of MACE stands for major adverse cardiac event. So they looked at who had a major adverse cardiac event over the next 6 years. And there really wasn't any difference. There was no particular outcome difference, partly because people stopped taking their omega-3. So by treatment group, there was no difference in omega-3, in MACE. But the fascinating thing that they did was they asked the question, well, what about people who increased their omega-3 index during that 6-month period by over 5%? People who really took the omega-3s and really got a big change in omega-3 index. Was there any difference in their outcomes compared to the people who didn't have over a 5% change? And here, what they found, here's the overall rate of developing a major adverse cardiac event. People who did not experience a big change in their omega-3 index, not more than 5%, their rate of MACE was 7%, but the people who did see a big change in their omega-3 index during that 6-month period, only about a 3% increase, 3% incidence. So that's a highly significant, basically a 59% reduction in risk for MACE for those who had an increase over 5% versus those who did not. And so and this is over the different years. The green is the omega-3s. It looks like they were lower after year 2, 3, 4, and 5 after they had been on this had the omega-3s during their healing process, they had a bigger change in the omega-3 index, they got a better outcome. So the achieving a target omega-3 index is more important than really being told to just take omega-3s. I mentioned Alzheimer's disease, and I think I'm getting out of time here. This is a study we did in Framingham. The higher the omega-3 index, this is risk for Alzheimer's disease, this is risk for dementia, and the odds ratio is 0.5, 0.6. That means a 50% reduction in risk for Alzheimer's if you had a high omega-3. We also looked in a previous study at the omega-3 index as it relates to the size of the what's called the hippocampus in the brain, the memory center, and we found that a higher omega-3 was associated with a increasing size of the hippocampus, so retain the structure of the brain better. I'm going to skip that one. We looked in the a big cohort called the UK Biobank, which is a big study being done in England, and the data are available to researchers around the world. We looked at risk for testing positive for COVID as a function of omega-3 levels, and saw that there was a decreased risk for actually testing positive, a decreased risk for being hospitalized with COVID, and a decreased risk for dying from COVID if you had an omega-3 index of about 8% versus about 3.5%. So confirming again our findings. Here's an interesting paper just published last week. Omega-3 levels were linked to reduced risk for hospitalization for sepsis. So sepsis, of course, is a systemic infection in your blood, terrible high-risk outcome. And they did in the UK Biobank, and they looked at individuals who developed sepsis as a function of their omega-3 levels, and they looked at quartiles of omega-3. And what they found was, compared to the lowest quartile, if you look at this model 2 here, the hazard ratio or the risk for being hospitalized with sepsis was 25% lower if you had a high omega-3 versus a low omega-3. So again, this has been seen over and over again. The omega-3 index is really related to better outcomes across the board. So to summarize, omega-3 index is a well-accepted marker of omega-3 status, superior to plasma-based measures, certainly on an individual basis. For some research purposes, plasma is probably okay. It can be determined on a dried blood spot. It can be used both in research and in personalized nutrition to help patients achieve an optimal omega-3 status. And the response to supplementation is variable, so testing is required. Current evidence shows that the index is a predictor of risk for premature death, for cardiovascular disease, for progression toward dementia, COVID, sepsis, and we can go on. This is, I looked at back on the citations and the research citations to our original 2004 paper. As of now, this is the number of citations per year. So it continues to go up. People are continuing to use this metric in their research. I did a Google Scholar search for the term omega-3 index, and it's been used about almost 7,000 times since that time. I was particularly happy to see this paper come out here this year in the Journal of Nutrition. Kevin Mackey is a longtime researcher, very well respected. He did a review of omega-3 polyunsaturated fatty acid testing in humans. He looked at commercially available options, and he concluded at the end of the paper, the OmegaQuant has the most widely used test with the largest evidence base, and that's the omega-3 index. And so that's a great place to be 20 years after we got started. And so I'll stop there, and hopefully we've got some questions that Tavis can lead us through.
Doc, thank you so much. Fantastic job. First question I think I can answer. I think I sent this to Christina. She was asking, how does the omega-3 index compare to the OmegaQuant? It's the same test. So we are offering the omega-3 index at OmegaQuant. So if you are using that, that's what you are measuring. Great question here by Maya. What are Mongolians eating in their landlocked country that gives them a better omega-3 index than Chinese?
Yeah, great question. I mean, right, this map, we took whatever published literature is out there. We don't know how well they analyzed the samples, but yeah, there was one report that looked at Mongolia, and it was pretty good. So no answer to that one right now.
Is there a plant-based omega-3 supplement for vegans, or is fish source simply the best? If so, what's a good fish product you can recommend?
Yeah, there are vegan friendly uh, omega-3 supplements. They're made from algae. For particular single-celled algae, you can find those widely on the market.
Why does the index not include ALA?
Because ALA does not increase the predictive value of the whole point of the omega-3 index is to predict risk for disease. And when we include ALA in a modified uh, omega-3 index, which we've done in a couple of studies just to ask that question, it doesn't, don't predict outcomes any better. So we stuck with the original marker.
How long a retrospective period does the omega-3 index measure? What period of time would be required to note a change after a significant ongoing dietary change to
Yeah.
boost omega?
Yeah, because it's measured in red cells, and red cells have a lifespan of around 4 months. We always say 3 to 4 months is a good time to recheck after you've made an increase in your omega-3 level intake, rather.
Um, I thought that you are encouraged to avoid oily fish and omega-3 supplements 24-hour before the omega-3 test, but based on what you say, okay, this is more of a statement. Red blood cell omega does not seem to be affected by acute omega-3 intake, which means you do not have to be fasted to take the omega index.
But yeah, that's generally the case, right?
In the Flux study, were intakes of other types of fats in this paper as well, and was it tracked?
No, but people were randomized to these groups. They were told to not change their diet at all. So what else they were eating shouldn't have been a factor here. Could have been, but we don't know. There's no reason to think because they were randomized, anything would be different between them.
Yeah, I know. I think you covered this, but can you talk more about the different factors within an individual that could increase the need for omega-3? You mentioned activity, they said age, sex, etc.
Yeah, probably older individuals might have more difficulty just because they tend to not absorb food as well as some of the enzymes start to fade away. So I think they might help. Um, we don't know. People who've have their gallbladder out might not absorb omega-3s or any other fats as well. That needs to be studied. But genetic differences, we really don't know.
Great question here. How do you explain the large variation in measurement between OmegaQuant and other labs? Were they using the same gas chromatography mass spec method?
Some of them do, some of them it depends on which fatty acids you put in the total fatty acid because it's a percent of total fatty acids, and if you only put 4 or 5 fatty acids in the total, then you're gonna get a different percent as omega-3. Some people don't, protect their dry blood spot cards with antioxidants like we do at OmegaQuant, and so when the sample is mailed in, the fatty acids degrade in transit, and it looks like you got a lower omega-3 level, simply because the, sample was not properly protected, so that's part of it too.
Do we need to take omega-3 supplements or consume food high in omega-3 for life in order to keep around the 8% range?
Yeah. It doesn't right, you don't get cured of a low omega-3 like you get cured of a cold. You gotta keep going.
Yeah, I had a colleague, dietitian friend that was at about 11%. He stopped. He just said, I'm going to do a little study on myself and stop for 3 months, went back down to 7% by not consuming omega-3.
Yeah, sure.
Is the amount of EPA and DHA or the ratio in supplement important in predicting the omega-3 index? What are your thoughts on that?
To the first part, yes, the amount of EPA plus DHA together is, kind of the good metric. We know that the higher that intake, the better. Whether it's all EPA, DHA, EPA, tends not to raise the index as much as DHA on a gram for gram basis. They both do, but not quite as DHA is a little better. So we like to emphasize that people take both EPA and DHA. They both have health benefits, and they both affect the omega-3 index. So as a general rule, we like supplements that have EPA and DHA. Whether it's stronger in one or the other is not that important.
Interesting question. How accurate is using hair strands as a measurement of omega-3?
It should be lousy. Never tried it because there's not really any reason that omega-3s would be in your hair.
Yeah, so this question we really can't answer just because there's a lot of great products and great brands. What omega-3 best elevates? I don't know if there's one that's the best, but a lot of the brands we work closely with that you see here are co-partners, Nordic Naturals, Yoli, um, um, Carlson Labs are excellent brands that all have great products in triglyceride form that will increase the omega-3 index as well. So one,
Right,
give it to
right.
that brands. Um, Dr. Tom Dayspring, Dr. Lipitos,
Tom,
great
hi,
job.
Tom.
Um, let's see.
I just listened to your talk yesterday with Peter. It was wonderful.
Are you concerned about all the sustainability of fish oil consumption with the recommendations for the ideal omega-3 index?
Yeah, I think that's always a concern that as we continue to decide that we need more omega-3, we're going to have to come up with other sources, which is partly why the algal approach is possible, is a good one. We're also doing we aren't people are doing research for land-based plants that, if they're genetically modified, can produce EPA and DHA, and that will be someday, I think, a solution for us too. That will not have to actually kill fish to get omega-3.
Just a couple more questions. It says Quest reports the omega check as EPA plus DPA plus DHA. Is it better just to measure EPA and DHA?
Yeah, it's the same thing for, as I said, for ALA. We looked to see whether DPA added any additional predictive value to EPA plus DHA, and it did not. DPA is a value that is not really that much determined by your diet. It's more metabolically determined.
All right, just a couple more questions for time. So if possible, we won't be able to accept any more questions after these last couple. How much extra omega-3 per day should someone take if they exercise regularly? Do we know that?
Obviously depends on where they start. We would typically say, and I think Kelly's going to talk about this later in the week, if you're around 4% and you want to get to 8% on average, something around 1,500 milligrams EPA DHA for a normal person. If you're an exerciser, a elite, really hard exerciser, I would say 2,000 to 2,500 milligrams probably is what it needs. And Tavis, you probably can answer that better than I can.
Yeah, I mean, on the performance side, we actually saw that study that came out of Baylor that showed they were giving 2,800 milligrams of EPA and DHA in a really good form, and levels only went up about 1.5 to 2% in 8 weeks, which is quite small. So personally, yeah, I mean, with athletes I've worked with, I've used in the 2,500 to 3,000, and sometimes high.
That's in a training during a season of sports, right? During that.
Correct. Yeah, and sometimes in the off season, but it's really good just to measure,
Yeah.
supplement for 2 to 3 months, and then see how it's impacting you. Last two questions. When processing a blood sample by venipuncture versus finger prick, is there any difference in the omega-3 results?
No.
And then let's see, trying to see if there's a question. Okay. Just more of a kind of a case study. I take 30 grams of flax for blood pressure, BMI, cholesterol impact. I also take a gram of fish oil 4 to 5 days a week. I eat 2 cans of sardines about twice a week and salmon when I dine out. Omega-3 index is 9%. Any issue you see above?
The flax oil is not doing anything for you. I mean, it's, from an omega-3 point of view, it's fine for other purposes, but it's all your sardines and salmon and your supplement that's doing it for you.
Okay. That's the last of the questions. Dr. Harris, thank you so much. Really appreciate your expertise for today's session. Thanks, everyone.
Thank you.